it. Okay. Anyway, hello all. I'm happy to be giving this lecture this afternoon, talking about biodiversity patterns in dragonflies. And interestingly, I can't move this. You see, there's never a lack of glitches.
I think if you click on it, it should go.
Oh, just click on it once and then it'll...
Yeah, and then I think, yeah.
Oh yeah, good, okay. So native biodiversity, this is a large group of insects, not as large as many of the really large groups of insects like beetles and moths and flies and wasps, but it's a respectable order with 6,323 recognized described species right at this moment, of which about half are damselflies and about half are dragonflies, and 4 are an intermediate group called damsel dragons, Anisozygoptera, 4 or possibly only 3 species. Just as a place here, diversity in one place, 462 species now known in North America, north of Mexico, in other words, the US and Canada. I can still see a fair bunch of people on the screens. I wonder if they could close their screens. Maybe I just don't need to look. And 443 in Peru. So I'll be talking about tropical biodiversity a lot. And so here's a country 6% the size of that US and Canada and has almost as many species. So that sort of gives us an introduction to the biodiversity in the tropics, which I'll be talking about. Nice to know that we are still very actively discovering and describing species of odonates. This is a graph from 2001 to 2020 of the number of species described in each of those years. I put this together for a paper in zootaxa, and that's why zootaxa is separated out at the top. As you can see, a very large number of species are continually being described, an average of almost 60 new taxa a year right now. And we've gone from 5,164 species in 2000 to almost 1,000, well, no, more than 1,000 species more in 20 years. And it looks like it's sort of dropping off a little bit, but I think there's still many undescribed species and collections, and surely many undiscovered species throughout the tropics still. The numbers above the bars are the numbers of new genera that were described in each of those years. So we're still reshuffling the taxonomy. Some of that is splitting up genera into several. Some of that is actually finding a species that is so distinct that it's actually a new genus itself. So I've taken part of a little bit of that, and really one of the high points of my life has been discovering unknown species and naming them. The one on the left discovered in Costa Rica in 1966. The one on the right I discovered in Washington State in 1971. And I still love seeing these species and some others that I've described whenever I can. Just an idea of the diversity of this group. You all know this, but here's some pretty splashy odonates, damselflies here from various parts of the world, tropical, pretty much all of them. That's certainly where a lot of the beauty in odonates is, except here's our ebony jewelwing, Calopteryx maculata, very common all through Eastern North America, just as beautiful as any of the tropical things as far as I'm concerned. And the same with the Anisoptera, a tremendous diversity of color and form and behavior. Makes them just such interesting creatures to study. They're diurnal, they're large, they're active. They've been called birdwatchers insects more than once. And they're really one reason we're so interested in them, because we see them around us. Fortunately, at this latitude, we see them only in the summer. But if we want, we can go dip in the ponds and get their larvae or nymphs out. And don't forget the Anisozygoptera and Epiophlebia, which lives only in Japan and has some relatives elsewhere in Asia. A creature that's sort of more closely related to dragonflies than damselflies, but it's a pretty distinctive creature. I've never seen it. I would really love to. You have to go to Japan in the spring. The larvae, of course, are even more diverse in shape than the adults are by far, really. The two on the top are both gomphids, the clubtail family, showing how incredibly different larvae or nymphs can be in that lived very different lives. The one on the right, an aphila, a forceps tail, lives buried in the mud, and the end of the abdomen modified into a long tube for breathing. It sticks it up into the water to be able to take in water and during respiration. The one on the left, Hagenius, the dragon hunter, is sort of mimics a dead leaf and lives in debris along the bank just below the surface of the river. And then a damselfly in the lower left, an enelagma, very, very distinct from dragonfly, the lower right, an emerald. So one of the things I've studied is alpha diversity, looking at the diversity at a single site. This is highest in the tropics, as I'll show you a lot more to come. And I chose, probably by accident, I was in Peru in 1977, and I ran into a birding tour in Lima. And they said, hey, we're going to go to Explorers Inn in the southeastern part of Peru. It's a brand new lodge that was just built. Nobody has been there yet. We're going to do a bird survey there. Would you like to come along? And so my wife and I went along for just a 3-day visit to this Explorers Inn, and just had a fantastic time and really concentrated on finding what dragonflies were there. I had a permit to collect dragonflies in Peru, so I spent an awful lot of my time looking at and collecting dragonflies, as well as looking at the birds and all the other neat things there. It has a known fauna now of 186 species at this one site, 186 species within walking distance of the lodge, the highest diversity of any single site in the world so far. It has varied wetland habitats, lakes, ponds, streams, big streams, little streams, rivers, marshy areas, really, no more varied probably than a lot of places in the temperate zone, but just a lot of habitats for a lot of species. These 3 days in 1977, I collected 200 specimens of 77 species. So these were the first, in 77, I got 77, the first records of any odonates even from that part of Peru, I think. Then I went back, my wife and I went back in 2002 and spent a month there. The first one was in June, the second one was in July, so pretty much the same season. And we collected 300 specimens of 119 species. But between those two visits, people had been staying at Explorers Inn, some of whom were friends of mine and colleagues of mine, and had been collecting dragonflies pretty regularly throughout that time. So what I know about it now is a combination of these two visits, plus many, many, many other specimens collected, mostly by non-specialists. They didn't know dragonflies particularly. They just had an insect net, and they collected and preserved dragonflies and sent them to me. Explorers Inn is in southern Peru. The equator runs right across here, south of the equator, way down in the southeast part. It's on the Rio Tambopata here. This is the lodge itself. The Rio Tambopata runs into the Rio Madre de Dios, which runs into the Madeira, which runs into the Amazon. So it's in the very uppermost Amazon basin, which is an area of extreme richness and diversity in many groups. This is a map of Explorers Inn and the trails thereof. The lodge is right up here in this corner. We went, most days we hiked from the lodge to Cocococha, which is a big oxbow lake. It was about a 5-kilometer hike each way, and basically looked for, collected and photographed dragonflies all along that hike. We also visited Laguna Chica. We went out through some of the forest trails. We visited Caticocha here twice, very similar oxbow lake to Cocococha, but we only went to it a couple of times. And the forest trails are and there are streams all along the forest trails running either into the Rio Tambopata or the Rio La Torre. So as we walked to Cocococha, we passed about 5 streams. So during that month in 2002, this is the accrual accumulation of odonatus species that we found. So pretty quickly, we got up to 20 species. Then we had a few days of really bad weather. They have friajes there that are cold weather that comes up from the southern hemisphere, from the south, just like a storm coming into North Carolina or something like that. And basically the odonates just shut down during that period. It was raining and cold and cloudy. And so we stayed inside or looked at birds. And then all of a sudden the weather got wonderful again. We started exploring more, and the species list just went up and up and up and up. We went to the Tambopata Research Center upriver for 3 days. So we're absent there, and then came back, and still pretty much every day we went out, we found additional species until we seemed to be reaching an asymptote or a plateau. And we had another day or so of bad weather here, but we pretty much reached this plateau of 120 species while we were there in a month. But it shows, this sort of graph shows that we probably have found most of what we were going to find when you have day after day of really not adding much else. This is a look at the same thing in a 25-year period, I think, from my first visit there in 1977 when I found 77 species. And then you can see pretty much people were present right on through this period. A lot of activity. A good friend of mine, Dave Pearson, was down there. He was actively collecting and getting other people to collect as well. And some Peruvians came out there. Gerardo Lamas and his students came out there. And so really a lot of collecting. All these were sent to me. And then probably not too much was being done during this period. And then some more collecting was done in this period. Then I visited there then and ended up with a total here. And then Heath Ogden and some of his students, I think Seth Bybee was one of them at the time, if I remember right. They went there again and added a few additional species for this total of 186. So again, we seem to have, we're getting there, but we probably haven't reached the asymptote yet. I would love to go back there for a month and see how many additional species we could find. So based on the specimens that I have, 1,851, averaging almost 12 specimens per species, this is the species abundance curve. So we've got a tremendous number of Urassus fastigiata, not only the ones I collected, but because it's such a very common, this is a wood skimmer, because it's such a very common species along all the forest trails, everybody that went there collected some. And so we ended up with quite a few of them. Argea caulata, a little dancer that's there, is the common Argea in most of the streams. And so most people that were there ended up collecting some of them as well, because they were along the forest trails as well as right on the streams. And so then from there, we go pretty rapidly down to species that you could call really very common. I'm not sure where a break would be from very common to not so common, but then out into a tail of less and less and less and less common species till the last bunch here, all this many species known only from one specimen each, this from 2, this from 3, and so forth. So a lot of really uncommon species, which is generally the case in the tropics. This is how it panned out if I looked at the total species per month. The dry season is from about May to September, and the wet season is from October to March. And the reason these two figures are so high, probably not because it was a dry season, but because those are the 2 months I collected there. And so all these were just general people trying to see what they could get. This was somebody knowing the odonates well enough to know if I saw something on the trail that I hadn't seen before, I better collect it. And so it may well be that the dry season is better for odonates only because it isn't raining so much. It rains a lot in the wet season. So that people are a lot of days when the odonates just aren't flying very much. But I would really like to have more information. I kind of don't want to go there in the wet season because they told me it may rain every day. And that's not for an odonatologist to love. But these would even out some probably. Just to show that you really need to collect a lot to find out what occurs in a place. Certainly that's true for a place that has almost 200 species. These are monthly totals of collected specimens and the number of species in each of those months. And you can see when you really, really collect a lot of specimens, you get a lot of species. And this is R square of 0.94, very, very high correlation. The more specimens you collect, the more species you get in a place that you're sampling like this. Here's some of the beautiful odonates we found there. These were odonates, 3 of 50 species that we found on the 2 coaches, Coco Cocha and Cati Cocha, the Oxbow Lakes. And sorry, I just screwed up on this. I didn't relabel. This is Protonura paucinervis. I copied and pasted all these names, and darn it, I didn't change that one. This beautiful thread tail is Protonura paucinervis. That's beautiful. Diastotops, very common, one of several species of amberwings that were there. Comparing the 2 coaches, we went to Coco Cocha more, but interestingly, we actually found more species at Cati Cocha. They were rather similar. The trail goes to the shore of the lake, and Coco Cocha, there was actually a dock and a boat, and we went out in the boat on that. Cati Cocha, we just basically had access only for a short distance around the shore. But they're pretty similar looking. They're similarly formed, similar vegetation, and yet this is so typical of the tropics. You go to 2 places that look similar, and look at that, only about half the species are in common. So this is, again, these sorts of things need to be tested more and more with more locations, but it's something I've seen myself in certainly almost everywhere I've been, certainly in Washington State and Florida, where I've lived, in North Carolina, two places that may look rather similar. You go there and you get pretty different, you can get pretty different odonates, which just shows the odonates know more about what they're doing than we do. These two places are really more different than we think they are from the standpoint of a dragonfly or damselfly. The swamps, there were quite a few swamps. A swamp is a wooded wetland. And in the high latitudes of Washington State or Massachusetts, there aren't a whole lot of odonates in wooded wetlands. The sun isn't, there's not enough sun, there's not enough productivity, there's not enough aquatic plants. So you really have a few specialists, but not all that many. Whereas you get into the tropics where even though there's not a whole lot of sun, it's still very warm, the water's warm, highly productive, and you get a tremendous number of swamp species. So these are just some of them at Explorers. And there were a couple of swamps along the trails that we checked very regularly. This beautiful Zenithoptera, the sapphire wing, was quite common in one of the swamps, one of my very favorite dragonflies, and a fire tail, a telebasis, and a wedge tail, the acanthagram, very common in these swamps. About 5 species of microthyria, these speckled dashers, and a rhodopygia, and this beautiful red Amazon darner. We're all in these swamps, and again, many other species, as you can see, for a total of 35. Now, not going again. Here we go. As I said, there were streams across the trails, and we sampled them. We actually would get off the trail and walk up or down the stream for a ways just to see what was there. And 26 species, not as many as in some of the lentic or the pond environments, but still pretty impressive, and almost most of them restricted to the streams, including these very beautiful, brightly colored damselflies. This is in the jewel wing family, and this is the Polythoridae, another beautiful family, and Paralestidae and the Argea caulata, the very common Argea there. There were 5 species of Argea though. Storaphlebia auca, one of the largest dragonflies of the world, cruising up and down the stream. Never could photograph it in nature, but we caught a couple of them. And these again are just sampled. The streams were interesting. You can divide it further than this. The streams were either muddy or sandy. The sandy ones were a delight to walk in. The muddy ones were a horror to walk in. I would sink up to my knees sometimes in those streams. I'd be really careful. I was afraid I'd disappear in the quicksand or some quick mud. But anyway, they were different. And so that's even another division we can go between. And we did find different species in the sandy and the muddy streams. This is a forest. And then so during our walks between these wetlands, among these aquatic habitats, we were of course in just beautiful rainforest. This is tropical rainforest at its best, with huge buttress trees and all the diversity of insects that you find there. And anyway, there were some odonates that pretty much always we found right along the forest trails, and these are just a sample of them. Of course, the helicopter damselfly, formerly Pseudostigmata, separate family, 4 species of this group were there, and they breed in tree holes. And this very rare orthomus, the largest orthomus in the world, and one that breeds just in tree holes, we found only a single one. This isn't my photo, it's Rich Hoyer's photo. Found only a single one of those. Very, very great thrill. Metaleptobasis, we don't have a very good idea where they breed. They breed in forests, and the nymph is undescribed. They're not very well known, but we found 6 species of them at Explorers Inn, 4 of which were undescribed, later described by Natalia von Ellenrieder. 5 species of Urasus, these wood skimmers were there. They were certainly the most common odonates along the trails. And then quite a few dusk flying dragonflies, Gynacantha, Triacanthogina, Nereshna, all these genera. We would flush them along the trail and try to see if we could catch them or photograph them if we saw where they landed again. And then at the station, at the lodge in the evening, all of these dusk flyers would come out and fly around and tempt us. And we caught as many as we could. A small one that flew at some height. We managed to jump up and catch a couple of them. They turned out to be an undescribed species. And so I didn't say anything about that. There are still at least a half a dozen undescribed species in the collections we made there, which I haven't got to. And there are another half a dozen that probably are described and I've been unable to identify yet. Well, and I noticed the forest had the largest number of species, probably because we spent so very much time in the forest. And of course, all of these species either bred in wetlands, either temporary or permanent, or up in the trees in phytotaumata and tree holes and such. So that had the largest list of any. And the forest is a great place for odonates in the tropics. Those that breed in nearby open wetlands often move into the forest to feed because it's pretty productive. Those that spend the dry season in the forest, there are those that spend the dry season in the forest and breed elsewhere in the wet season. So they may breed in open marshes and ponds in the wet season, but they move into the forest in the dry season because the humidity is greater there, the insects are greater, and it can be really dry and hot out in the sun. So better to get into the forest and cool off a little bit and be able to find more to eat. And then there are those that breed in the forest itself, in the swamps, in temporary pools, and in tree holes. When the rainy season comes, so the people there told me, the trail itself gets flooded in some areas up to a foot deep. And even in the higher areas, there are little pools all along the trail. And that's where some of the wood skimmers, the Urassus breed, and some of the damselflies, and the big darners, the big dusk flying darners, all breed in those pools. So the forest is of great importance in the tropics. And the fact that it's being cut down at such an incredible rate is endangering odonates as much as birds or mammals or anything else. We studied Urassus wood skimmer behavior. I had two students there, one of whom was surveying damselflies on streams, the other of whom was actually marking Urassus and studying them. And I worked with her all through it, but a lot of this was her finding. And she marked individuals of the 4 species. There in lower right is a Urassus imbuta with a paint mark on the wing just to alert us to the fact that it was numbered. And then she actually numbered the wings of each individual that she caught and marked. So she had several hundred individuals caught and marked along several of the trails. Really, a really good, neat thing. She found that they were, and now this is the dry season for these dragonflies. Most of them probably not breeding. They were essentially immobile. She would find them at the same place along the trail day after day after day. And I was helping because I would record where we saw her marked individuals. She saw virtually no feeding. She would watch them for many, many minutes at the time. And basically they would just sit, and they weren't, and I'm sure they must have fed some, but very, very inactive compared to what we often think of dragonflies being. And she looked very much for mating and egg laying, and found none except a few individuals, Urassus siemensii, the one species she actually found, breeding. So really interesting study. Here's the 5 species of Urassus that were there. They all have a pseudo-ovipositor. The subgenital plate is extended, very much like a spike tail, a corduligaster in the northern hemisphere, for pushing eggs into the mud. So the females hover over shallow water, and even mud before it gets inundated with water, and push their eggs into it. And then of course, when it does get inundated, the eggs hatch and the nymphs develop and emerge and so forth. But look at the interesting variation in the 5 species that were there, from a very short subgenital plate to a very, very long one in Urassus ovipositrix. And presumably they're doing something different because they weren't really breeding at the time. She really didn't know any, and we still don't know anything about what Urassus are doing. Really interesting genus, needs more studying. So this is just another interesting thing. This is a place I actually spent much more time at the Hacienda Taboga in Guanacaste in Costa Rica. I spent 14 months there actually studying odonates, and I hope to publish that before I die. I feel really bad that I haven't. I've written some small papers on it, but the whole big study really needs to be written up and published. So anyway, this is just an interesting thing about it. I studied 5 different marshes. I named them all there within walking distance of where we were staying in some cabins. And and I studied them throughout a year. So I mean, I really knew a lot about them. And so they had sort of similar numbers of species, but the 2 at the bottom had more species, and this is the total that I recorded. And then surprisingly, they only had about half of the species in common. So again, these marshes that were grassy or cattails, dried up, all completely dried up in the dry season, flooded again in the wet season, and were inhabited by lots of odonates, had an amazingly different fauna. And I couldn't tell the difference between some of them, and yet the dragonflies apparently could. So let's go away from explorers in and look at some larger scale patterns, area diversity relationships and odonates, diversity patterns within the US and world patterns of sorts. And I think I actually don't have any world patterns. I think I just pretty much restricted it to the New World, and tropical diversity and comparative diversity. So here's the thing, island biogeography has received a lot of study over the years, and people say that larger numbers of species live on larger islands, and we know it for a lot of creatures, but it's had never been looked at for odonates as far as I know. So these are West Indian and East Indian islands, number, size of island versus the number of species. And it's a pretty good relationship, just as it is in birds and reptiles and amphibians and some other groups that have been studied. The larger the island, the more species. The biggest one at the right is Borneo, biggest East Indian island. The biggest West Indian island is Cuba here. And here is a fairly strong relationship in both cases of island size and number of species. Just more possibilities for different habitats on bigger islands, less possibility of species going extinct because there are too few of them on a very small island. I looked at odonate at country size and diversity. Anybody would say, of course, the larger the country, the more species it should have. And that's certainly true with Brazil, almost off the chart here, with a huge, huge South American country with a huge, huge mid 600 numbers of species, and then down to a whole bunch of other species, which on this graph are not too easy to figure out. So I made a graph with lacking Brazil. Can do a little bit better job on these now. And you can see this is the line that leads to Brazil pretty much. And above these line are species with especially large species lists, or countries with especially large species lists, and probably needs to be explained why Venezuela has so many species for its size. Is it just because it's had several resident odonatologists there? Might be as simple as that. Yet Mexico has resident odonatologists, Colombia does. These other countries, not so many, but Argentina has a lot as well. And so Chile and Argentina, of course, are getting more into the temperate zone, and they're well below this curve. And these other countries are somewhat above it. And this is all the smaller South American and Central American countries sort of spread around a lot, enough so that you get almost no really nice relationship at all in this graph. So a lot of interesting things going on that relate to diversity of habitat, that relate to both ecological and almost surely historic reasons for having the size fauna as they do, as well as have they had resident odonatologists, how much collecting has been done there. Look at odonated diversity in the US. Now I have to say that I first made up this talk to show at the University of Puget Sound when I was on the faculty there, and I retired 15 years ago. So some parts of this are from actually that far ago, about 16 years ago, but still I have, they're pretty much the patterns I think are pretty much the same. The range map on the left I made up for river jewelwing, Calopteryx equabilis, and here's the map on the right from Odonata Central. 20 years ago, maybe I made up this map on the left. You can see we do know more. We have more records for Wyoming, we have more records for Montana, but still, even 25 years ago, we had some reasonably good idea. And of course, these are some of these maps are the ones that I made up that I used for my North American field guides. And we've learned so much more since then. I wish Princeton would let me revise the field guides, if only the maps in them. Here's an interesting thing, odonated diversity in Washington, my state. I live in Seattle, right about here. And these are the county lists for all the counties of Seattle out of the 83 species known from the state. And guess what? 60 in King County here is the highest. It's because I live here. So this sort of shows you something about the way we learn about diversity very often sort of radiates out from where the person lives who studies it. The closer to home they are, the more species they find. This is a very rich part of the state. The whole northern edge is sort of, the boreal forest comes down here, very wet, very rich, lots of odonates. This is the second highest number. I've spent a tremendous amount of time there. This is the county. This is Yakima County, where Clarence Hamilton Kennedy lived for a while and wrote one of the first papers about Western Odonata, really wonderful paper on Washington and Oregon and then California and Nevada. And so he's found a lot as it is. And then I've spent a lot of time in that county just revisiting a lot of his area. But these counties are kind of poor. These counties are largely agricultural. They do have wetlands, they do have ponds, and marshes, but they really don't have as many species as some of these more wooded species. And then species like this are isolated on the Olympic Peninsula, very wet much of the year, not the greatest odonate habitat. So I can actually relate a lot of these numbers to, number one, what the physiography and the climate of the state, and number two, where I've spent a lot of time. Spent a fair amount of time in Little Wahkiakum County here, not many wetlands and very rainy. So maybe that's why it only has 19 species. And I got to click again. So here's some diversity of back then, this was 15 years ago, I compiled the county list for every county in the United States. And I don't know where that little line just came from. Did I draw that? Oh, no, back. Anyway, can you all see this line that goes across there? Maybe. Oh, you're muted, so don't worry about it. You're muted. Anyway, this is I compiled county lists for all the counties of the United States. Okay, this was a giant job, and now it's easy to do on Odonata Central. Just go on Odonata Central down, but there's enough counties that I just did not have time to do it again from Odonata Central. So this is somewhat old, but still, it makes the point that the highest county lists were somewhere in the neighborhood of over 120 species. And this is where this is the mean latitude of the state that county had the highest list in. Okay, and so what I tried to do is see if there was any latitudinal pattern in diversity in the United States, and there really isn't. There's except that the North seems to have even higher county lists than the South. Could this be because some odonate people have been living in these places for a long time? Not necessarily, because there's some counties in the South that have had odonate people in them for many, many years. So not a very good correlation. But if you try it another way, and I'll click again. If you separate the United States into West, Central, and East, you get some interesting patterns. In the central part of the country, there's a really strong latitudinal gradient of R squared 0.9. That's extremely strong. So Texas here has a lot more species than North Dakota, and it's almost a straight line. So you really get a decline in species numbers. And again, this is not the state list, it's the number of species, the highest county list in that state. So it's a pretty good measure of diversity. So and then in the West, you get something like that too. Not anywhere near as strong, but the northern states, unfortunately, I'm in one of the northern ones, doesn't have as many species. So there's something different going on in the West. In the East, it's really, really different. You get not a very strong pattern at all. And the diversity actually rises a little bit in the North. So why would this be? Oh, and the other thing I did was looking at longitude. And so the highest county lists and the mean longitude. So this is backwards. The West should have been in the other direction, but I didn't figure out how to do that. So you can see there's a lot more odonate species in the East than there are in the West. Even the rich states like Arizona and so forth don't make it to most of the New England state. The highest county list in Washington state the Washington state list is about half that of Rhode Island. So there's really, really a difference in the United States between West and East. And here's some measures I did again, and these would be all larger now. There'd be more counties with these colors. But I also looked at counties with over 100 species, which they were. There was one in Florida where Minter Westfall and his students lived, one in this part of Georgia, and a bunch in North Carolina where Duncan Kyler collected for years, and then scattered all around the Northeast, these really, really high county lists, nothing at all in the West. And the red is not quite as high, but still a really high county list. Remember, the highest county in Washington was 60, lots of those in the East. And not just in the Southeast, but all the way up to Maine. And even in Southern Canada, there's probably places of equal diversity. So really, the East really has the odonates. So let's look at two variables. Temperature, it's warmer in the South, odonates like warmth. It's nice and relatively warm in the West, but this is an annual temperature. Our summer temperatures in Western Washington along the Pacific coast here are much lower than the summer temperatures at the same latitude in the East. So not the best odonate country. And let's look at the precipitation. The West, the Pacific coast is very wet, but this wet area correlates with low summer temperatures, so really not such great odonate habitat. In the east, it's plenty wet all the way up into New England, very good for odonates. And so sort of the odonate diversity sort of almost follows this pattern. You get a lot of wetlands, you get a lot of odonates. But the interesting thing is to look at the historical aspect of it. During the glaciations, the glaciers came down across North America, drove all the vegetation zones south, drove all the animals and plants south. But the vegetation zones and precipitation were continuous from the east into Mexico. So all the things that might have evolved to live in this area at that time did just fine in the south. The things that evolved to live in this area did just fine in Mexico. And then as the glaciers retreated, they could just move back up into their original range, which we know so many plants and animals did, including surely dragonflies. In the west, on the other hand, the glaciers drove all these species to the south, but there was a huge amount of arid country, much lower precipitation. So the glaciers actually made a lot of species extinct because they couldn't do all that well moving into this really dry area. And then there really wasn't that much of a fauna to move back up. And I think that explains a lot of the patterns that I've just showed you here, this particular pattern. So let's go into the tropics again, where it's warm and wet and couldn't be better for odonates pretty much all year, although some areas have pretty prolonged dry season. And tropical and Zygoptera are what are really, really diverse in the tropics. Here's a Euphia species from Malaysia, a Diphlebia species from Northern Australia, Protolestes from Madagascar, a Rhinocypher from Borneo, and a Heteragrion from Brazil, or from Venezuela, this is. They're just some of the absolutely wonderful damselflies that we have in the tropics. And so we look at damselflies, and the present phylogeny now gives us 33 damselfly families and 11 dragonfly families. Much, much more diversity at a higher level, at a higher family level of damselflies than there are of dragonflies. It used to be about the same until this new molecular work has split up the damselflies into more and more and more families. If we look at the percentage of a given fauna that are damselflies, in the US and Canada, it's below 30%. So below 30% of the odonate fauna are damselflies in the US and Canada. Much, much lower in Chile, which is, of course, a very temperate country. But then as you move up closer and closer to the equator, you get a higher and higher proportion of damselflies, so that Colombia and Ecuador have a very high proportion, 50% or more damselflies. So notice the damselflies are what really increase with this increase of odonate to the tropics. And so finally, let's look at some comparative diversity. This I thought was really interesting. Graphing some creatures against one another. This is a graph of freshwater fish diversity against odonated diversity. And so these are freshwater fish state totals on this axis, odonated state totals on this axis, just some indication of diversity. And it looks like there's it does go up with freshwater fish diversity and odonate diversity go up, but it's a pretty poor correlation, r squared 0.32. And that's just because there's just a huge amount of variation in all these states. There are some states that have a relatively high freshwater fish diversity, not so many odonates. Some states that have a relatively high odonate diversity, not so many fish. This is presumably something that has to do with the great difference in all the different states, some better for fish than others because of different kinds of wetlands. But anyway, they're both aquatic. So there should be some relationship, and there is at least some relationship that you can see. But you compare odonates with a group that are not aquatic, mostly birds, and there's just no correlation at all. These are country totals for odonates and birds. Dragonflies on this axis, birds on this axis, going up here to 1,700 plus species in Colombia, but only 300 odonates. And again, I know this number would be larger now. And that's kind of interesting too. There's not really a very high correlation at all because they use different things. Odonates need fresh water. Birds have all sorts of other needs. And then I compared two areas of somewhat similar size and pretty well-known faunas, Costa Rica and New York. This is a taxonomic group here. This is the ratio of Costa Rica to New York. So Costa Rica is about half the size of New York. but it has almost as many fish species as New York does. It has more odonate species, quite a few more odonate species, and you can see how it goes up. So these are aquatic. So there really isn't as much difference in aquatic faunas, and this is something I've seen because I've been interested in both terrestrial and aquatic faunas all my life. They're really, both fish and odonate sort of need the same thing. So there really isn't this tremendous tropical diversity gradient from the temperate to the tropics that you get in birds, mammals, reptiles and amphibians, and butterflies. The butterflies are so much more diverse in the tropics than in the temperate zone. And it'd be interesting to do this for other groups as well. So whatever, these are warm-blooded animals, endotherms, the herps and the butterflies. So they, they should do better in the tropics. And odonates are too. Why aren't they like butterflies? Well, because they live in fresh water, and that gradient is different between freshwater and terrestrial species. Terrestrial species have a tremendous amount more opportunities because they're not confined to water. I looked at odonate species richest individual locations. These are the highest county lists of all these. You've sort of seen this already. But here's some tropical localities down into Mexico: 133 species in the Tuxtlas, 47 species on the coast of Quintana Roo, and some totals for Costa Rica: 98, 89, 91, all places that I studied in Costa Rica. Couldn't quite reach 100 species in any of them, but still quite rich. And one in the Panama Canal here. And then down into South America, highest lists in individual places: Limon Cocha in Ecuador, the island of Trinidad here, places in Western Brazil, and then Explorers Inn here, which actually, I did this before the total went up to 186, and the area north of Explorers Inn, and then one place, Iguazu area of Argentina. So no place seems to be able to get very much above 100. Well, we get pretty large numbers right here in New England, though, comparable to some areas almost in South America. So that, again, that gradient is there, but nowhere near as great as it would be in birds or other terrestrial groups. So some conclusions: odonates are distinctly more diverse in the tropics. That diversity gradient is strongest in damselflies. Species diversity is related to habitat diversity, as well as basic abiotic factors of like temperature and precipitation and type of wetland, say. There's a much greater array of odonate habitats in the tropics because they use phytotelmata. They breed up in the trees. They breed in wooded swamps, which is not a great habitat for them in the temperate zone. Both ecological and historic factors are important. The movement of the glaciers were important. Changes in South America has gone from much wetter to much drier over historic times. That surely affected odonates. And of course, we're seeing that in real time now with climate change. We're seeing how droughts are having severe effects on odonates. They dry up wetlands that would otherwise be used by dragonflies and damselflies. And at the opposite end, extreme rains clean out, flush out streams completely, scour them out. Pretty much, I know of several streams in Costa Rica now that have had so much rain from hurricanes that have pretty much lost their odonate fauna. They'll probably get it back at some point, but you can really see these severe effects of the present day situation in odonates very clearly. And there are different patterns and different taxa, both within odonates and between odonates and other groups. And I think that's the end.